Thank you, Mathilde, for the introduction. And thank you all for showing up here in person at UCSB, but also online via Zoom. So for this talk, I'm very excited to tell you about geometric intelligence in minds and machines. So both the work that is being done in our lab at UCSB, but also the research from collaborators around the world. And we're going to talk about minds and machines. So this is going to be at the intersection of neuroscience and artificial intelligence. And the main question for this talk is going to be, how do intelligent systems encode information about the world? So, for example, our brains have 80 billion neurons, and somewhere in these neurons is encoded what I see, what I hear, what I touch. Large language models also have 8 billion parameters, and they can memorize what you told them 20 days ago. How is this information encoded and stored in intelligent systems? If we look at brains first, and we look at the data may look like this. So here in this animation, each row represents one neuron, and you have a white dot every time that neuron is spiking. So each row has what we can call a train of spikes, a spike train. And somewhere in this apparent random patterns of spike train is the information about what I perceive about the world. And not only what I perceive, it's enough for me to make decision about what I perceive and really act in this world. So where is this information encoded? These spikes are very intriguing. So now we have only 3 spiking neurons, say. They're very intriguing because they are binary signals. The neuron is either on or off. So it's binary, and yet what I feel, what I perceive, feels very continuous to me. So I can tell the brightness of the light, I can tell the loudness of a sound, or the intensity of a touch. So how can a binary signal on or off be able to encode this seemingly continuous information? Someone who gave a first answer to this question is Edgar Adrian, won the Nobel Prize in 1932, and he designed an experiment that looks like this. So he took the leg of a dead frog, and in that leg, he recorded from one neuron. So he stuck an electrode to record from one neuron. That neuron was known to spike when the muscle of the frog was being stretched. And so he attached different weights to the muscle of the frog, so 50 grams, all the way to 500 grams, to see how these neuron spiking trains will change when the muscle is stretched more or less. Did you read that? Yeah. Sorry, Chase, we put you on the other screen. You can just collapse it too. Yeah, perfect. So he recorded what happened to this neuron, which is a neuron within the muscle of the dead frog, as this muscle is stretched more or less. So more stretched when we attach more weight to the muscle. And he wanted to see what happens to the spike trains. And what he saw is that the magnitude of the spikes doesn't change when we increase the weight that is attached to the muscle, but the frequency of the spikes does, or the rate of the spikes does. So the heavier the weight, the higher the rate of spikes. And so that's what is now known as rate coding. This is how neurons that can either be on or off can encode continuous information. So now we have not only the first neuron, but let's say 3 neurons, and each of them now has a continuous information attached to it, which is the rate of spikes. Kind of answer the question, how does one neuron encode information? But it doesn't answer the question, how do these billion neurons work together to encode information? And so that's where geometric intelligence comes in as an answer to this question. How do these 3 neurons, or the billions of neurons, work together to encode information? And the picture of geometric intelligence looks like this. Here we have what we call the neural state space. It's a vector space where each coordinate represents the rates of one neuron. So you have neuron 1 on this coordinate axis, neuron 2 on this one, neuron 3 on this one. In this simulation, it's a 3D space because I'm looking at 3 neurons, but generally it's an 80 billion dimensional space. And the main idea behind geometric intelligence is to say the geometric patterns that emerge in this 80 billion dimensional state space is how information about the world is encoded. So it may seem like a crazy kind of artistic idea, right? What would the brain draw some beautiful and mesmerizing patterns to encode information? But what's even more crazy is that this has been observed in natural brains and artificial brains. So I'm going to give you a few examples of that. And first, geometric intelligence in natural brains. So this is an experiment by Garner et al. That was published in Nature 2022, so it's fairly recent. In this experiment, an animal moves freely in this 2D space. So this is a 2D environment where the animal moves, and it's going in 5 seconds from this point to this point. And it is aware of its 2D location, we can say. Gardner and colleagues recorded from 149 neurons in the brain of this animal, and then draw the rates of firing of these neurons in neural state space. So neural state space is now 149 dimensional, because we're recording for 149 neurons. And this is what they saw. Even though the space is 149 dimensional, there is a 2D structure that emerges and that looks exactly like the torus we had on the simulation. And superimposed on that torus, you have the trajectory of the rat. So now not in 2D space, but in neural state space. So at the beginning of the trajectory, the rat is here, and we can read from here the firings of its neurons. And as the rat moves through 2D space, then there is a corresponding trajectory in its inside world that is lying on this torus. So that's already, to me, exceptional. That's something that could have been 149 dimensional, is actually 2-dimensional, which, by the way, is the same dimension of the 2D space in which the animal is moving. But what's even more mind-blowing to me is that somehow I would have expected a 2D plane here, because the rat moves in 2D space, and that is a flat surface. So if we're talking about a 2D manifold that emerges in neural state space, I would have expected a 2D plane here, but no. Somehow the brain decided it was more efficient or more optimal in some sense to encode this 2D environment in this 2D torus. So there might be something optimal about that. If there is something optimal about that, then maybe AI will converge to the same strategy. You know, brains and evolution of brains has converged to encoding 2D space with torus. If we train a neural network, would we see the same thing emerging? So let's do it. We and others have done it. So now you train a recurrent neural network to move in 2D space and to predict its own position. So you're sure that the RNN knows where it is. As input, you give it its velocity, and as output, you ask it to predict where it is in 2D space. So the same square, and you let it train. And then you stick an electrode, a virtual electrode, to record from its neurons, hundreds of neurons. And if you do that, what do you see? A torus emerges in neural state space, and this has been reproduced in a series of work. So what is so particular about this torus that it emerges both in brains that have been evolved over millions of years and in RNN that we can train in a few minutes on the computer? These are the two tori that I was just telling you about. But now more and more work are beginning to see other types of manifolds emerging both in natural brains and artificial brains from different regions of the natural brains and in response to different stimuli. There is a really nice literature review by Chang and Abbott from 2021 that summarizes what has been seen, but I'm going to show a few. So first, in natural brains, across the animal kingdoms, we see these geometric structures appearing, these neural manifolds, they call them. across the animal kingdom for the animals in which we can stick electrodes inside the brain. So fly, bats, rodents, and mouse. And I'd like to point out two that I really like, starting with the fly. So here we're recording from the brain of a fly, not from all of the neurons, but just the neurons that are called the head direction cells. So these are neurons that are there to record the direction of the head. So as the fly is flying, there is a group of neurons that records where the head of the fly is pointing. So you could imagine, well, that's an angle, basically. So it makes sense to be encoded in a circle in the inside world of the fly. And that is actually what happens. You can record from 15 neurons from this fly's brain, you're going to see a one-dimensional circular structure. And what's I think even cooler is that you can look at the same group of neurons, but now in a bat's brain. So still the head direction neurons, but not in the fly, now in the bat. And the bat tends to do more crazier things as they fly. And it turns out they don't only record the direction of the head as parallel to the ground, but also the direction of the head, like pointing up or down. And in the case of the head direction cells or neurons of the bat, we see a torus that emerges in the neural state space. That's for the examples about the natural brains. In the artificial brains, we see nonlinear manifolds or geometric structure happening, arising. And here you can tell that, especially when we give them images as input, so if you give images as input to artificial neural networks, you have manifolds that emerge, but are less well characterized somehow. It's not only rings of torus, it's like more complicated shapes. And so part of the research we do in our lab is try to design tools that can characterize these shapes and relate them to how the intelligent systems encode and act in the world. Okay, so until now, I've told you about geometric strategies that are being discovered in brains and machine, encoding the head direction around a circle, encoding 2D space on a torus. But in parallel to this research community, you also have kind of the pure deep learning community, who is not necessarily interested in brains first, but whose goal is to develop better and better AI models. And in particular, there is the geometric deep learning community, topological deep learning, and equivalent deep learning communities who are employing geometric strategy to design better neural networks. So without necessarily looking at what is happening in neuroscience, they're thinking, oh, maybe I can put this geometric constraint here or this topological constraint there. And they design architectures that tend to perform better on ML tasks. So now that we have this neuroscience field, we can ask the question, could the geometric strategies that have been developed in geometric deep learning, topological deep learning, equivalent deep learning, actually exist in the natural brains? Because if they are so great, and if that's where deep learning should go to get better models, then maybe evolution already found that solution. So we're going to look at two kind of communities. First, topological deep learning, and how we have indication that this is actually happening in brains. And then equivariant deep learning, and a few indications that we have that is actually happening in natural brains. Starting with topological neural network. So this is a literature whose aim is to generalize graph neural networks by encoding higher order interactions between neurons. So it's artificial neural networks, it has neurons. These neurons can pass messages between them, and you can encode more and more complicated interactions in order to pass the messages. Our lab did a survey of this literature if you want to learn everything about it. Let me tell you about the interaction between the neurons. So you could design a neural network where the neurons do not interact. There is no weight connecting them, they don't send messages. We don't do that as much. Then you can design a graph neural network that looks like this. So your input data will be a graph, nodes and edges, and the data will be on this graph by features on the nodes and maybe features on the edges. So imagine maybe your data is an atom, and on the nodes you have the type of atom, and on the edge, the distance between the two atoms. A graph neural network would process this data layer after layer to make a prediction, maybe predict what is the molecule, what is the property of the molecule. And it does that by leveraging interaction between atoms or interaction between nodes through something that we call message passing. So to update the features from layer to layer, the nodes will send messages. What's important for our purpose is that these messages only travel on the edges. This is why we say that graphs leverage pairwise interaction between a pair of two nodes. And now what topological neural networks do is design more complicated interactions between neurons so that you can design more complicated neural networks. So here we still have 6 neurons, but now neurons can be related not only by edges or pairwise interaction, but maybe 3-way interaction or 4-way interaction. And I also have different shades of pink because maybe some interactions are stronger than others. So you can think of this as a very general framework to encode all types of interactions. And then you get neural networks that look exactly like the graph neural network from before, but now messages can move all over. These neural networks, as we show in the review, are shown to outperform graph neural networks. So it seems to be a good strategy to encode information about the world. If it's that great of a strategy, does it happen in brains? So what I'm going to show you is real data, not from our lab, so I don't have any stake in this, from Maoz et al., published in PNAS in 2020, which I think is convincing evidence that natural brains do topological deep learning. This is real data from 17 neurons recorded from the visual cortex of monkeys. And the goal of this paper was to model the probability distribution of the spikes. And they did that in 3 ways. First, they assumed that the neuron spikes, but they don't interact. Second, and again, it's their figures, not mine. They assume that there is a pairwise interaction between the neurons. And third, they assume that they have higher order interaction. And I added the combinatorial complex thing. But this is their figures. And with each of these models, they compared 2 things. The probability distribution of the spikes of the neurons, more precisely, probability distribution of patterns of spikes. Some patterns that happen not that often, some patterns that happen more often. So that's what you have on the x-axis. The real empirical probability of patterns of spikes. And here you have the ratio of probability predicted by your model and the real probability of this pattern of spike. So if your model matches the empirical observation, this is 1 and this is 0. That's what you want. Okay, so how does it work when you have no interaction? Works like this. So you want to be here at 0. Here in this gray zone is the 99% confidence interval, because you're not sampling from these neurons forever. So you have kind of this sampling fluctuation. You're not going to have the exact same probability between your model and the empirics, even if your model is perfect. So that's the 99% confidence interval. With no interaction, the data is here, so we're not winning the 99% confidence interval. With pairwise interaction, already it's much better. And you see that the points which represent each patterns of spikes, they tend to be closer to 0. The probability of this pattern of spikes predicted by the model versus empirically observed starts to be the same. But what's even crazier is that if you add a combinatorial complex, now all of a sudden you're, again, much, much better. So that seems to be first evidence that topological deep learning is a good strategy also for natural brains, in this case, for monkeys. Second example, equivariant and invariant neural networks. So this is another side of the literature where deep learning researchers have incorporated geometric strategies in neural networks, both with equivariant and invariant artificial neural network. So on the left, equivariant neural network. On the right, an example of invariant neural. This neural network is called equivariant. Here, for example, it's a neural network that extracts the gray scale version of the image. It's called equivariant for the group of rotation, say, because if I transform or if I rotate the input image, the output is rotated as well. So it's equivariant in the sense that the output changes in relation to the input. And the neural network on the right, by contrast, is called invariant because it doesn't change its output when the input is modified. So here we have an invariant network whose goal is to predict what's the object. The object is a hand, and it doesn't change. It's invariant if the hand is rotated. And through a series of works that we also review in this recent literature review, it is shown that this type of neural network tend to outperform classical neural networks. So same question, if that's optimal for deep learning, does it exist in brain? Did evolution find that solution to the learning problem before we did? So I'm going to show you, again, real data, not from our lab. And there is this literature review by Higgins and colleagues that also gives you more examples of this. This is a schematic illustration of the brain that's looking at images, say, maybe images of the hand. And what first activates is the primary layers of the visual cortex that we call V1. And that's where you have edge detectors and so on. And then the information flows into two pathways. One's called the dorsal pathway and one that's called the ventral pathway. And what's really fascinating is that the dorsal pathway is also called the where stream. And that's where location and transformation is encoded. So that's where equivariance happen. If the hand is rotated, then that's going to be rotated too, or the rotation is going to be encoded. And last but not least, the other one is called the what stream, and that's what records the shape of the object, the shape of the hand. If you rotate the hand, the shape doesn't change, so that's where the invariance is encoded. And that's an example of an experiment made in 1984, which was quantifying this ventral pathway that is invariant to transformation. So they show different images. I forget to what type of animal, but to an animal. The little arrow shows you when the stimulus is presented, and the x-axis is the time. And you can see that this neuron is basically encoding for the shape of the hand. If you rotate the hand, the neuron still fires, and it fires across also different way of drawing the hands. This is exciting. Now, what our lab wants to do is go beyond and ask, can we push these ideas even further? So far, evolution was there before us. Brains have evolved to do topological deep learning, equivalent deep learning. Can we push beyond what evolution has created? I'm going to give you one example, which is an example where we've put complete invariance in neural networks. You can think of it as a stronger notion of invariance, stronger to what that pathway was doing. And the goal was to say, let's take the MNIST, actually the rotated MNIST data set, which is kind of the #1 benchmark in machine learning, and see if we can predict from each image what's the digit of that image, and if we can do that with the smallest neural network possible. So think of it as a DeepSeek type approach. We don't want to do that with a giant neural network. We want to do that with the smallest neural network possible. So we're doing one convolution, possibly several layers, one pooling, and then an MLP. And we want to see in this convolution layer, if we decrease the number of convolutional filters, will it still perform well? Well, unsurprisingly, if you decrease the number of convolutional filters, this CNN accuracy will drop. So that's why we build big AI models, right? Usually it doesn't work when you remove all of the convolutional filters. In our approach, we leave the convolutional alone, and we agree that we're going to remove filters, but we're going to change the pooling operation that comes after. Pooling operation hasn't gotten much love in AI for some reason. Everybody, use max pooling. We replace that block by another computational primitive. And this is what happened to our model when we decrease the number of convolutional filters. The accuracy stays the same, and in particular, with as low as 2 filters, we have an accuracy that's similar to what we have with 20 filters. So that's kind of a strategy to build better, more efficient neural network by pushing these geometric ideas to the limit. All right, so concluding, in answer to this original question, how do intelligent systems encode information about the world? Our answer is with geometry. And that's what could have been surprising at first is not that much surprising if you think about the history of physics. In particular, in statistical physics, people had, similar problems in that they needed to model the interaction of atoms. And they solved that with statistical mechanics. And so similarly, I would like to argue that when we want to understand what millions or billions of neurons are doing together, we need a dedicated theory. And for us, this theory is called geometric intelligence. So these are kind of the main pillar of our labs. And I want to thank the students, the postdocs, and all of our lab members for making this theory a reality. And thank you very much for that.
In the interest of time, we'll do one question, and if you want to speak to Nina afterwards, go for it. So, does anyone have any questions here or on Zoom? Yeah.
How does the pooling operation differ from max pooling, and is there a theoretical intuition as to why it preserves rotational?
Yes, great question. I was kind of hoping someone would ask that question, so thank you. It's called a complete invariant, and that's the completeness is the main difference with the max pooling. So max pooling is like you have an image and you take the maximum intensity of its pixels. So if you have a dog, you take the maximal intensity, maybe it's white somewhere in the head of the dog, that's the maximum intensity. You rotate that image, you take the maximum intensity, well, that's invariant rotation, because okay, the pixel has moved, but the intensity of the pixel hasn't changed. So max pooling, finding the maximum intensity is invariant rotation. But in the process, you've destroyed all the information about the dog. Because I can create a random image with the same maximum intensity. Max pooling is going to give me the exact same information. So what we've designed with a group extension of Fourier theory is an operator that catches everything about the image, why that's invariant to rotation, while preserving everything else. So for us, we're going to keep everything about the dog except its orientation. So that's how we're more robust and complete. Thank you.
Thank you, Nina. Rapidly moving on to part 2. I am very happy to introduce our second speaker. Louisa Cornelis is a second-year PhD student in the physics department here at UCSB, where she's a member of the Geometric Intelligence Lab that we just heard about. She holds undergraduate degrees in physics and computer science from Scripps College and Harvey Mudd College. Luisa's research lies at the intersection of machine learning and biology, focusing on leveraging geometric deep learning methods to analyze proteomics data for biomarker discovery. She's also interested in fairness and model explainability, working to ensure equitable and interpretable applications of machine learning models. Luisa is supported by the National Science Foundation GRFP Fellowship. Thank you, Luisa.
Great. Hello, everyone. Thank you all for coming on Zoom and in person. So I'm going to continue this talk by kind of showing an application of a lot of the things that Nina was talking about and explaining decoding frontotemporal dementia, proteomics via graph neural networks. And this work was done in collaboration with lovely collaborators at UCSF and the Memory and Aging Center, and then also within my lab here at UCSB. Okay, so show of hands, who here knows what Alzheimer's is, has heard of Alzheimer's? Okay, great. Now, who knows what FTD is, frontotemporal dementia? Besides my lab. I feel like you guys have heard about it. So way less people. And so let's learn about FTD and how it's different than Alzheimer's. And that makes sense because FTD accounts for approximately 10 to 20% of all cases of dementia, but it's still obviously very important. So let's learn about it. So FTD represents a group of brain disorders caused by degeneration of the frontal and/or temporal lobes of the brain, as illustrated here. So it causes changes in behavior, personality, language and/or motor skills, and then deterioration in a person's ability to function. And we don't currently have any prognostic biomarkers for FTD, so there is no cure similar to to Alzheimer's. So how are Alzheimer's and FTD different? So they have different symptoms. As I just said, FTD brings a gradual progressive decline in behavior, language, and/or movement, and memory is usually initially spared until later stages in the disease. So as opposed to Alzheimer's, where usually one of the first symptoms is not remembering things, that actually isn't as common in FTD. It strikes much younger. So it's the most common form of dementia for people under 60 years old. and the majority of FTD cases occur in the 45 to 64 age range. So because of the age at which it occurs, it really has a great impact on family life, economic status, and all these other things that happen, because usually people aren't retired yet and maybe they have children. Also because of the symptoms and the age at which it strikes, it's often initially misdiagnosed as a psychiatric problem or movement disorder. So a lot of the initial symptoms are social cues, actually a lacking of social cues. So one thing that actually is really common that we see at the Memory and Aging Center is inappropriate comments towards women or about people's bodies or things. And patients will come in, they'll go to their doctor, and people are kind of like, well, you just need to control that. But actually, it's these fundamental changes that are happening to someone's brain that are only recognized many, many years down the line when it declines and progresses a lot further. And also, it has a much stronger autosomal dominant genetic component than Alzheimer's. So what this means is that there are a few key mutations, genetic mutations, that if one of your parents has that, there's a 50% chance you'll inherit that gene. And that means if you live long enough, you will 100% develop this form of dementia. And that accounts for about 20% of all FTD cases. And in Alzheimer's, that number is much, much lower. So I just came back from 3 months doing a research rotation at UCSF and was actually able to go to a lot of the clinical visits and meet with these patients. And it was really impactful to see, you would see people come in with their parents who had one of these mutations. And a big question for them was like, how do I manage this uncertainty? Do I want to get tested? Do I not? Just generally, how do I handle this? And so while it is a rare disease, it most likely has affected people you have heard of. So Bruce Willis has frontotemporal dementia, as does Wendy Williams. This article is about a software engineer, Lee Holloway in the Bay, who started developing symptoms at around 30 years old, and no one really knew what it was until about 10 years later. And he actually got treatment at UCSF. And these two articles are more about the genetic component. So people who know that one of these genes runs in their families and are trying to grapple with that and understand what that means for them. So how can we better understand frontotemporal dementia? Well, a huge answer to this is proteomics, which is the study of the entire protein composition of an organism. And this has recently been transformed by new scientific instruments. So similar to how 30 years ago the Human Genome Project emerged with the goal of completely sequencing the human genome, which is about 30,000 genes and is now complete, proteomics is the new wave of this. And proteins are responsible for a wide range of functions essential to life. So every process from metabolism to immune response is controlled by your proteins. Unlike genomics, protein expression changes over time. So it actually provides us a really unique window into disease progression and like when someone shifts from asymptomatic to symptomatic. Something that makes this interesting but also more difficult is that the proteome is estimated to be at least 3 times larger than the human genome. So that's a lot of data. So how do we comb through all this? How do we make sense of this? And because of this, biomarker discovery remains a major challenge. And a biomarker is a molecule that signals whether your body is functioning normally or experiencing disease. So it's a really good target for therapeutic treatment or just for doctors to monitor how your disease is progressing. And we don't really have any of those for FTD yet. And in this talk, I'm going to specifically focus on expression proteomics, so measuring the concentration of your proteins in your CSF, so cerebral spinal fluid or plasma. So what are some current methods that we're using on these data sets? One really common one is differential abundance analysis, which is a mass univariate statistical method used to identify differences between different groups in your data set. So you might get a plot like shown here with this volcano plot, which shows, okay, say we have a data set of carriers and controls, and we want to understand what proteins or genes are really important. We look at the abundance ratio between these groups and the statistical significance. And as one of my collaborators, Rowan, always says, is that when we're doing these analyses, like the way we find biomarkers is they really just like slap us in the face on these plots. And they're going to be the points that have the largest abundance difference and are the most statistically significant. But this is also looking at each protein as an individual contributor to disease. And in reality, we know that the underlying biological processes are much more complicated. Another common method is lasso regression. So similarly, models proteins as individual predictors of disease outcomes and doesn't take into account any of these higher order relationships. And you may say, okay, well, we can add interaction terms, but if you have a data set of 7,000 proteins, adding interaction terms between all of them is not really feasible given the sample size. So your model is no longer reliable. And also it might force some of your main effects to become zero. So that's also not reliable. And another key thing about both these methods is that they're linear. So they don't take into account any of these nonlinear relationships that could exist. So how do we solve this? Well, based on Ina's talk, you can maybe guess where this is going. We use graphs. So this is an image from another paper just showing like a general protein interaction graph. But like I said, we know that proteins work interactively in the regulation of biological processes and disease progression. So we're going to leverage this interprotein information and format our proteins into data set specific graphs. So in this case, for a data set of frontotemporal dementia mutation carriers and controls. And we do this because while we know that there are fundamental biological pathways, we also don't know how those change as the disease progresses. So we want it to be specific to this disease to understand it better. So what are the goals? The first goal is to predict how can graph neural networks improve disease severity prediction using large-scale proteomic data? And then also, how can we use these networks to discover new things? So how can these networks provide insights into the molecular underpinnings of FTD? So there's been a lot of amazing work being done in this field. With regards to the data set we're using, our collaborators at UCSF have done a bunch of large-scale proteomics analysis using the linear methods that I talked about earlier. And then in terms of GNN omics, there's also a lot of really cool work emerging in this field. But something that's important to note is that studies combining machine learning and proteomics are still relatively rare. So only about 2% of recent proteomics papers include machine learning. So among these GNN omics papers, only one of them has been applied to a proteomics data set, and it was more of a proof of concept on a smaller COVID-19 data set, and none have been used on this FTD proteomic data set. So that's what we are doing here. So overview. First, we need to format our graphs. How do we want to structure this data to capture the information we need? That's important. Then, how do we use these graphs and apply an explainable, biologically inspired graph neural network to it to predict these hallmarkers of disease progression? So that's either going to be NFL or cognitive slope, which I'll talk a little bit about. And then once we have our network that's performing well, how do we understand what it used as important in this prediction to understand new biomarkers or pathways that are contributing to this disease? So I'll start with formatting these graphs. So what data set do we have? The data set was collected by the All-FTD Consortium, which is a nationwide consortium. So thank you to them for allowing us to use this. It includes cerebral spinal fluid proteomic data characterizing 7,258 proteins. Within that, there's also an average of 3 annual visits evaluating cognitive status. So patients are seen by a neuropsychologist. And then we also have plasma NFL measurements. And NFL is a biological marker, which itself is part of the axonal cytoskeleton. So as a neuron becomes more damaged, more of that is released into your plasma or CSF. So it's a really reliable index of progressive neurodegeneration, and it's currently the gold standard for FTD, but it's important to note it's not FTD specific. So for example, if you were to get a concussion, you would have a higher rate of NFL in your blood because of that brain damage. So it's not the ideal biomarker, so that's why we're still searching for more. And our data set is around 230 people, carriers and controls. We have 3 mutations, C9, granulin, and MAPT, which sounds small for machine learning purposes, but is actually very big for the medical world. So first, we apply weighted correlation network analysis. So we take all of our protein data across all of our subjects, and we get, for each pair of proteins, we calculate the bi-weight mid-correlation. So we want to understand how correlated is each pair. And then we apply a soft threshold to this to reduce noise. And then we apply a hard threshold to gate whether we're going to have an edge between the nodes in our graph. And so each of our nodes is going to be a protein, and an edge tells us, okay, are these sufficiently correlated to deem a relationship so they can communicate as they pass through our network. And we want to pick our soft and hard threshold values such that there are a few nodes that have a lot of connections, but the majority have very few. Then we have this overall structure, and this is shared across all people, right? So we found that for the whole data set. So then we have to format each person within this graph structure. So we take the graph structure that we had, and on each node, we put their specific protein expression values. So we have around 240 graphs, and on each node are there specific measurements that capture where they are in the disease. Okay, so then we have to take this graph and use a GNN. So from here, I've been talking a lot about how we want to capture all these higher order relationships. So how do we do that? We use graph attention layers to construct multi-level graph features. A graph attention layer uses an attention mechanism to dynamically weight and aggregate features from nodes. So like Nina was explaining, you can sum over your neighbors, but this actually, the network learns which neighbors are the most important in that final output. So not only are they connecting all of them, but it's also learning in a dynamic way. So we start with our G1, which is like our initial graph with protein expression data. And then we apply a singular GAT layer, we get G2, and then we do that again to get G3. And those are our hierarchical graph features and more like protein module level features that are capturing these module level and then even greater level relationships, which are all really important for omics representation learning. From there, we also incorporate graph patient level features, so sex, mutation, and age, knowing that those are also critical in how diseases progress and our bodies work. So we take all of those features, all our 3 levels of graph, and we pass those into readout layers. And we can predict NFL or cognitive slope. And so we explain what NFL is, which I'll show you the results for that here today. Cognitive slope is kind of a future goal down the line, which is a measure of how much someone is cognitively declining from those cognitive exams that I talked about. So it would be cool to see like this clinical marker and also this biological marker and see what the differences of emerge as important pathways for those both. So in terms of our results, so it's important to note this is all preliminary work, so they're single runs, and we did some benchmarking, but experiments are currently in progress to use different seeds and do like a proper k-fold analysis, make it more rigorous. But so for context, this is a histogram of NFL, and the values span from 0 to 120 picograms per milliliter. So if you run lasso regression, you get a root mean square error of about 13.15 picograms per milliliter. If you run just a normal deep neural network, you get 10.31 picograms per milliliter. And then if you run our GNN, you do substantially better. So you get about 6.28 picograms per milliliter as your error. And you can see the actual values versus the predicted values on the right there. So it's really exciting to see that this information, like Nina was talking about, is actually important in improving prediction and how these diseases really manifest in the body. So lastly, once we have our network predicting really well, we want to understand what went into that prediction that made it so good. What relationships were then learned? And how can we leverage those to better understand the biology, the underlying biology? So we have to interpret our model, and we do this using explainability techniques. Specifically, we use a method called integrated gradient. So it represents the integral of gradients along the model from an output to an input. So on the right there, you see like a toy image. So say this is the landscape of NFL prediction, and then P1 is protein 1, P2 is protein 2. you're gonna have two people with different values for those. So you take a straight line from a baseline to those people, and you take the integral of the partial gradient with respect to each of those proteins to understand how much are these proteins contributing to this final result. And we picked integrated gradients because it's stable. So it's not a learned method, so we'll always produce the same result, which is really important for clinical purposes. It's efficient, and it satisfies sensitivity and implementation invariance axioms. So sensitivity means that if the input and the baseline differ by only one feature, but produce different outputs, that feature should have a non-zero importance. And then implementation invariance is that if you have two different networks that take the same inputs and have the exact same outputs, their explanation should also be the same, because we want to reflect true behavior rather than design works of our models. And so what do we get out of this? We get basically an importance score per person per protein. So this is subject specific. And another thing to explain is that, like, what does a positive importance score versus a negative one mean? So if something is positively important, it means that as it increases, NFL will increase. And if something is negatively important, it means that as it increases, NFL will decrease. So how do we think about this on like a person-specific level? So what we're looking at here is, we'll take patient 1, who's a MAPT mutation carrier, and patient 2. And if I look at their most important proteins, then I take the 200 most positively important for each of them. And then I plot the graph structure that was used in our network. This is what we see. And these nodes, the size of them is actually indicating how important those nodes were in the final result. So larger means it emerged as more important. And I'm going to point out some cool ones we see. So in the MAPT mutation, we see LRFN2 and MPTX2. I don't know all the names because I'm not a biologist, but so I'll just read them. And we know in MAPT that there's profound synaptic loss that drives neurodegeneration. And MPTX2 and LRFN2 are synaptic proteins that seem to be emerging as really important. And there are studies that have found that neuronal contraction 2, MPTX2, is actually a really important biomarker in FTD. And that's just something that our network found. So it's really exciting to see that. In our granulin mutation, we see C3 and TLR1. In this mutation, there's animal studies that suggest a very strong immune-mediated disease effect. And these two proteins are key proteins in innate immune pathways, which is the way of your body's response to biological stressors. So once again, there's some like biological grounding of what's emerging here. And I think it's important to note that without our collaborators at UCSF, this would not be possible because they really have this fundamental knowledge and understanding that allows us to make sense of these results. So that was on an individual level. So now if we want to look at the data set as a whole, what these plots are showing is if we take all people and their importance scores and we sum those all up and we take the mean importance of each protein across each person, what do we see? And on the right of both of these plots, you see the importance scores, so what I was just describing. And on the left, you see the differential correlation scores. So what would emerge in traditional techniques, not taking into account all these relationships. So one important one is chitinase 1, and we can see that that's really important for differential correlation and from our network. And that makes sense. It's a microglial protein that represents brain inflammation, so we expect that one to be important for both. But then we once again see all these proteins emerge that we wouldn't have found otherwise. So B4-GALT3, it's a sugar metabolism protein, and that pathway has been linked to neurodegenerative diseases before, but never with FTD. So it falls actually into this glycosylation alteration, which has been found important in Alzheimer's. For the negatively important, we see DYN-LT1 emerge, which is a cytoskeletal protein, and that's affected by what's called a tau mutation carrier, and we know that within our data set, MAPT mutation carriers will have this. So it's also cool to see it emerge here, despite not being seen with differential correlation. This is another way to visualize a very similar thing, but on this left plot, we see both of the points, locations of points on the plots and axes are exactly the same. It's the coloring that's different. That might help clarify. So the x-axis is each protein's regression coefficient with NFL, and this y-axis is the statistical significance. And on the left, those points are colored by mean importance, and on the right, they're colored by differential correlation. So chitinase, which was important for both, is really at the top. We would have pulled them on out with traditional methods, but DYNLT1 and B4GALT3 are really at the bottom. So clearly our network is learning these intricate relationships that wouldn't have been picked up by traditional methods. So once we have all these important proteins across everyone, we want to understand, okay, also what pathways do those contribute to? And so we can run something called the gene ontology analysis. And this you're seeing is, once again, on the top 100 most positively important and on the top 100 most negatively. And so what you do is you give this database a list of the 100 proteins, and the z-score represents how much higher than chance would having this many in a certain pathway be. So like a higher z-score means it's very unlikely that if you just gave it 100 random ones, you would have that many that fall into these pathways. And it's important to know also that there's thousands of pathways. So it's cool to see that it narrowed it down this far. So once again, on the left, we see these key pathways for inflammation. So tumor necrosis production and apoptotic signaling. And then on the right, we see metabolic processes. So synaptic signaling and metabolism. So really how well neurons are maintaining their energy and communicating with each other are key ones that emerge as important. So once again, the biology, the real point is the biology is lining up and is making sense with things that we would expect to see. Yeah, so in summary, we use biologically informed graph neural networks, and we surpass traditional techniques to predict markers of neurodegeneration. We identify proteins that are crucial for disease severity prediction that traditional methods miss, and show that graph-based deep learning on high-dimensional proteomic data reveals novel molecular insights. And this can hopefully be applied to other neurodegenerative diseases in the future. And then we also identify patient-level graph features that could tell us patient-specific epicenters of disease in the future. And really the next steps here are to figure out also how to translate these things into clinical applications, because the goal is really how can we best help people and deploy these findings into the real world to make the reality of having one of these mutations something that is not so negative. Yeah. Thank you, everyone. Thank you to NeurIPS and NSF for sponsoring this work.
That was wonderful. We have time for some questions if anyone has any, or on Zoom. No questions? Oh, yeah.
Sorry, you said it by accident.
Yeah.
How were these initial edges determined in the first place?
Sorry, say it
How
again.
were the edges determined in the first place?
Oh, so we found the bi-weight mid-correlation between each pair of proteins, and then you saw threshold that, and you then hard threshold that to decide if you want to have an edge between them.
Sorry.
Yeah, no.
It's biologically.
No, so the one way to construct the graph is to use these protein databases and just say, okay, we know these are in the same pathway, connect them. But this is actually data set specific. So it's just using the values that we have and finding the correlation between them.
Maybe it's kind of similar question, but how extendable is this framework to other omics data? So we transcript omics data, things like that.
Oh, yeah.
Do that.
Yes, it's super exciting. You could really apply like the same exact methods to it. Which is exciting. And you can, future work could be also using different types of omics data, neuroimaging, cognitive status, all these types of things to predict better. Yeah.
Obviously, for co-occurrence, it would be a lot more computationally intensive, but do you expect, like, if you were to include multi-way, like topological information between like triplets or higher order interactions or higher order co-occurrence,
Yeah.
would that?
Yeah,
Is
that's,
anything seen made you think that?
That's a great question that I was just talking about yesterday, actually. Yeah, so I think one of the key issues with that is just space,
Yeah.
because this data set is so large, and that's something that has been a challenge. But I think it would be really cool in the future to apply some of those higher order relations. And I mean, from what we've seen in previous research and studies, I have reason to believe it would help. But it hasn't been done yet.
Great, let's thank Louisa one more time.